Hello everyone, welcome to BioScan. This is Mohini Sharma, your biology facilitator. Today we'll be doing fundamental unit of life, which is a chapter of grade 9 biology. Fundamental unit of life. So what is the basic fundamental unit of life? Every one of us know that it is cell. Okay, now how was cell discovered? Cell was discovered by Robert Hooke in the year 1665. How did he discover? He was observing some cork tissue. Cork, you all know, which is the outermost portion of the stem, oldest stem, the bark. Okay, so it is a dead tissue. He was observing that. Now, when he was observing, he found that some hexagonal structures are there, like that of beehive. So he gave them a term cellulae. Okay, and this cellulae was shortened a little later, and then finally it was called as cell. So even the term cell was given by Robert Hooke. So who discovered cell? Robert Hooke. Who gave the term cell? Robert Hooke. Then there was one more scientist. See, because I told that cork is a dead tissue. So that means he discovered or he observed a dead cell. So who observed the first living cell? It was Anton von Leeuwenhoek. Or of course the name is a little longer and it is difficult, so you can shorten the name. It is A.V. Leeuwenhoek. Now we have seen that cell is called as structural and functional unit of life. This is the basic definition, that cell is the basic unit of life. It is the fundamental unit of life. That's what the chapter's name is. Or it is the structural and functional unit of life. So the third sentence becomes one of the very important questions for your exams. The first one, that why cell is called as structural unit of life. So why it is called as structural unit of life? Because all living organisms, whether you talk about unicellular, small, big, multicellular organisms, plants, animals, any organism existing on this earth is made up of cell. That means you take out human, you take dinosaur, you take elephant, you take a rat, you take a mango plant, you take grass, amoeba, they all are made up of cell. So every organism is made up of cell. It is the building block of the structural organization. So we call it as structural unit of life. Next question arises, why do we call it as functional unit of life? It is called as functional unit of life because it is the smallest entity. Entity means because it's a thing, right? But we cannot say it's not non-living. So we use a better term for that. That is, it's an entity. So it's the smallest entity that can perform all the life processes. Please mark my words. What I am saying is that can perform. It does not perform. So if I take an example of amoeba and a human, in amoeba, it is only one cell. That one cell performs all the functions. But in human, if I take one skin cell, does it perform all the functions? No. We have different type of cells performing specific functions. So that's why we say it can be called, it can perform all the life processes. For this reason, it is called as functional unit of life. So till now, what all we have studied? A quick revision for that. Cell is the structural and functional unit of life. It was discovered by Robert Hooke in the year 1665. First living cell was observed by Anton von Leeuwenhoek. You can even write A.V. Leeuwenhoek or just Leeuwenhoek. All are marked correct usually by the teachers. Then we have discussed about that why cell is called as structural and functional unit of life. Now, has anybody seen cell? Few of you must be saying yes, few of you must be saying no. So how to visualize a cell? See, if you can visualize a cell, it becomes really easy. So wherever you are watching this video, wherever you are sitting, imagine that particular room as a cell. Okay? So whichever room you are sitting in, that is a cell now. So the walls, these walls of the room becomes your cell membrane. This hard wall becomes your cell membrane. All the things here. So I have my entire setup. Okay, I am there, and other stuff is there in this room. Maybe for you also, many things must be there apart from you. Okay, you including other things become all the cell organelles and nucleus. So first thing, what I said is cell membrane. We all become the nucleus or the deciding factors. And then we have air in this room, right? We have air in this room, everyone has. So that air becomes cytoplasm, okay? Now you'll say, why are we, comparing this to our room? Okay. Think of it that what is the function of this wall? This wall provides this protection. Yes. It does not allow outside things to get in easily, correct? Then it gives a shape. So my room can be, circular, oval, rectangular, hexagonal, square, depending upon the construction of wall. So it gives us protection, shape. Then it helps in entry and exit. In this room, every one of us has a door, right? Every one of us have. So what is the function of the door? Certain things can get in and out. Why do I use the word certain? Can a car come in? No. Why? Because the size of the door is little smaller. Can elephant move in? No. Again, because the elephant is very bigger in size. If it has to move in, it has to break it. So this entry and exit is dependent on size. 3 functions we have done of wall. Okay. So first one, protection, shape, entry and exit. The third one, suppose if I have a window and somebody is just passing by and need to say hi to me. So that person need not enter my room, neither I have to go out of the room. That person simply can say hi from there and I can reply hi. Simple. That means nothing entered the room, nothing went out of the room, but the message was passed. So passing of message. Okay. Now this I compared it with cell membrane. That means function of cell membrane becomes protection, shape, entry and exit of molecules. Just an adding term. Entry and exit. Entry means coming in, exit means moving out. So entry and exit of molecules. And what was the fourth one? Passing of message. So this is how you can visualize a cell. Okay, the second component was we all, right? The things you. Now here I am the deciding factor. I will see how my room is arranged, right? What light should be off or on. The AC should be working or no. It is me who is the deciding factor of the room, correct? So that becomes the genetic material. What is the function of the genetic material of the cell? It controls all the activities of the cell. What is the function of genetic material? It controls all the activities of the cell. The third component, which is again very important, that is air. Okay. Now imagine, let us not take gravity into account firstly. Okay. Imagine that there is no air. And gravity, I said, don't take it into account. So what will happen? Nothing will be on its place. Like space, all of us would be flying here and there, right? So the major function of air could be to fill the place first, and second, to keep everything in its own positions without any movement. Okay. Third function, if I speak and there's no air in between, will you be able to hear me? No. So air is a transporting medium, right? Similarly, in cell, what would be the transporting medium? Cytoplasm. Now the 3 things that I have told you about cell. Okay. Cell membrane, which was this. Genetic material, which was me or you in your room. And the third one was air. Okay. These are the 3 essential components of a cell. For example, if I wish to call this as a cell, if I want to say that this is a cell, okay, how can I say? If I'll tally it with a checkpoint, okay? If it has, does it have a cell membrane? Yes, okay. Does it have cytoplasm? Okay. Does it have genetic material? Okay. If these three things are there, it is a cell. Out of these three, if any one is missing, it is not called as a cell. So I'll just repeat and summarize. 3 essential things to be present inside a cell: cell membrane, cytoplasm, and genetic material. What is the function of the genetic material? To control the entire cell, control all the activities of the cell. What is the function of cytoplasm? To keep all the cell parts, or you can say a better term, organelles, in its own place, and it acts as a transporting medium. And what is the function of plasma membrane or cell membrane? Plasma membrane is the another term for cell membrane itself. So what is the function of cell membrane? Protection, shape, entry and exit of molecules. And the last one was passing of, don't write high, it is passing of message. Now we have understood that what is a cell, what are the basic components of a cell. So how many shapes does it exist? It must be existing in so many shapes, right? So there are very small cells, very big cells, very small as in you cannot see through your naked eyes, very big, that means you can see through your naked eyes. Then they are spherical in shape, they are rod shaped, okay? They are comma shaped. You put commas while writing. So they are comma shaped. So we have a specific class, like those who are spherical, we call it as coccus. What do we call it as? Coccus, C-O-C-C-U-S. Rod shape, like rod, they are called as bacillus. What are they called as? Bacillus. And the third one that I said, comma shaped, they are known as vibrio. V-I-B-R-I-O, vibrio. So vibrio means comma shape. Coccus means spherical, and rod-like is bacillus. These are the different shapes of a cell. Any idea which is the largest cell? I guess most of you must be knowing that is an ostrich egg. Okay, then which is the smallest cell? This is a little tricky. Okay, the smallest cell is PPLO. PPLO full form is Pleuropneumonia-like organism. The other name is Mycoplasma. This is the smallest organism existing on this earth. And then we have longest cell. Longest cell is neuron or nerve cell. That is present in us also. It could be up to 1 meter. Imagine 1 meter. So they are quite long cells. Then do you know any cell that does not have a nucleus? A eukaryotic cell, what I meant to ask is, any eukaryotic cell that does not have a nucleus? It is mature RBC. Red blood cells, you must be knowing, which are there in our blood, they do not have a nucleus in it. Okay, they are non-nucleated cells. Rest all our body cells have a nucleus because they are eukaryotic, so they should have a nucleus. Now, what is prokaryotic, eukaryotic? We'll do it in the end of the chapter so that it becomes very clear whatever terms we are using there. So next topic is cell theory. Cell theory was basically given by 2 scientists, Matthias Jakob Schleiden and Theodor Schwann. I know it's bigger names, but what you can do is you can just remember Schwann and Schleiden. Now it's easy. Okay, so it was given by Schwann and Schleiden. What these both said is that cell is the basic unit of life. What we have already understood, right? Cell is the basic unit of life. All organisms, whether plants or animals, are made up of cell. Third point, these are the 2 major postulates. Postulates are the statements given in this theory. So 2 postulates, important one we have discussed. The third postulate was actually given by a third scientist, Rudolf Virchow, V-I-R-C-H-O-W. Rudolf Virchow, he said that all cells arise from pre-existing cell. In his language, he said, omnis cellula e cellula, which eventually means, of course, all cells arise from existing cell. Now, what do you mean by this? First, I'll tell you a short story about Schwann and Schleiden, and then I'll explain you about Rudolf Virchow. Schwann was actually a botanist. I hope you all know what's a botanist, those who research on plants. Okay, so he was a botanist, he was researching on plants, and he gave cell theory only for plants. Schwann and Schleiden are not friends or not connected to each other. In some other place on this earth, Schleiden was also doing research, but he was doing research on animals. So that means this person gave a cell theory for plants, that is Schwann. Schleiden, being a zoologist working on animals, he gave cell theory for animals. Eventually, in the later part of their research, they merged and formulated one theory, that is cell theory, which we know it today. So Schwann did it in 1838 and Schleiden did it in 1839, 1 year later. After 16 years, that's 1855, Rudolf Virchow, after analyzing, he said that one postulate should be more there in the cell theory, which was all cells arise from pre-existing cell. What does it mean? That cell do not arise spontaneously. Spontaneously means on its own. Okay, so through air and all, a cell cannot be formed. For a cell to be formed, there should be an existing cell. I can explain you in terms of humans very easily. If a human has to be formed, it has to come from a human only. Right? Simple. Human gives birth to human. Okay, now you're sitting here, air is there, light is there. Can you make a human there? Through by Baba Sam Chamatkar? No. Okay, that does not happen. So that means all human arise from humans only. Similarly, we can say this is for naturally. Don't ask other things like test tube baby and all. Okay, this is naturally process I am talking about. So what I was telling is that all cells arise from pre-existing cell. So these are the three major postulates of cell theory. Cell is the basic unit of life. All plants and animals are made up of cell. These two were given by Schwann and Schleiden. The third one, that's the last one which we remember, is all cells arise from pre-existing cell. And the third postulate was given by Rudolf Virchow. Now we'll understand the difference between unicellular and multicellular organism. Of course, by the name we understand uni means one, cellular means cell. So made up of one cell. Multi means many. Again, cellular means cell. So multicellular made up of many cells. Unicellular organisms are made up of one single cell and multicellular are made up of many cells. Unicellular basically does not have division of labor. Now, what do you understand by division of labor? See, division of labor is we have 5 people and one function has to be performed. Okay, one dance has to be performed. So what we people do, suppose it was my group. So what we 5 people do, we divide the work amongst ourselves. Suppose I will decide the song, somebody else will decide the choreography, third person will do the lighting and all, fourth will do the costume and all, and fifth will do music editing and all. So we have divided our work. By this, what happens? Efficiency is there, okay, and it takes lesser time. So same thing, that is called as division of labor, work, division of functions, division of work, which is absent in unicellular. Why? Because it only has one single cell, but in multicellular, division of labor is there. So we have a specific cells, group of cells that perform the function of, heartbeat, like it beats, okay? We have specific group of cells performing exchange of gases in lungs. So we have different organs for specific function. That means division of labor is present, but in unicellular, division of labor is absent. Unicellular, as it has single cell, suppose there is any injury or death of one cell, there is no other cell, so the entire organism dies. But in multicellular, if my one cell dies from here, will it affect me? No. So one single cell injury does not affect the, life of an individual in multicellular. Fourth point we can write for unicellular is that an organism is exposed to environment from all the sides. Because it has only single cells, so it is exposed to environment from all sides. But a multicellular organism like us, like me, it is not exposed. The cells are not exposed. Like my inner body cells are not exposed to the environment. Only my outer cells are. Correct? So this gives you more protection from the environment. Any example for unicellular, anyone? Amoeba, Paramecium, Euglena. These all are unicellular organisms. A tough question now. Give me any two examples of multicellular organism. Multicellular organisms, of course, humans. Please don't write plants and animals. Everybody know plants and animals are multicellular. Give name, right? Mango, pineapple, human, cat, dog, donkey, monkey, anything. Okay? Okay. So our next topic is cytoplasm. Do you all remember what was cytoplasm? Okay. So what is basically cytoplasm? It is the packing material present inside the cell. Okay, it consists of many substances like salts, then many fibers could be there, many organelles are there in it. Cytoplasm basically is the site of biochemical reactions. All the reactions happening inside the cell, actually majority of the reactions occurs in cytoplasm. And few functions we have done while I was discussing with a room-like structure. Okay, so it acts as a transporting medium. That means the sound can travel here, air. So in cytoplasm, what can? Anything that has to be transported within the cell, it will be done through cytoplasm only. Fourth, that cytoplasm keeps everything in its place. That nothing should roam here and there, nothing should jump, collide with each other. Okay, so it keeps the organelles in its own places. So I'll just quickly revise what is cytoplasm. Cytoplasm is a jelly-like substance present inside the cell. It is the site of all biochemical reaction. It is a transporting medium within the cell, and it helps in keeping all the organelles in their own places. Now the next topic is plasma membrane, which we have done. A little more we have to do. Before doing plasma membrane, let's understand certain terms which I usually love to give the heading as terminology. There are few terms which are related to cell, or you can say plasma membrane or cell membrane. So we should be aware of these cells. Usually in exam, they are asked in the form of questions or fill in the blanks, or directly asked definitions also. Okay, so very important. The first one that we are going to do is diffusion. I hope you all must be have heard of it. What do you mean by diffusion? Diffusion is movement of molecules from higher concentration to lower concentration. It happens on its own. I'll give a very easy example. Suppose somebody passes through this room and has used a very nice deo. Will I get the smell? Yes. How? Did that person blew the deodorant towards me? No. It came automatically. Why? The molecules there were more, here was less. So movement of molecules from higher concentration to lower concentration is called as diffusion. Second, osmosis. Now what is osmosis? Osmosis is movement of solvent. It is not movement of molecules. It is movement of solvent from a region of its higher concentration to its lower concentration. Here the word its is very important. I'll tell you later its importance. Okay. So its higher concentration to its lower concentration through a semi-permeable membrane. Now what is semi-permeable membrane? You'll get to know. Okay. So I'll just repeat what is osmosis. Movement of solvent from its higher concentration to its lower concentration through a semi-permeable membrane. Fine. Now what is exoosmosis? Usually, see, in osmosis also usually the solvent is water. Okay, you must be knowing that water is a universal solvent. So usually the solvent is water. Exoosmosis, that's the third term. Exoosmosis is movement of water outside the cell. So if this is a cell, movement of water outside is known as exoosmosis. And if movement of water is inside from outside, that means water is entering the cell. What will we call it as? Endoosmosis. In exoosmosis, what will happen? If water is moving out, the size of the cell will shrink. And if water is entering, depending upon animal and plant, it may change. So that we'll discuss later. Okay. Okay. So this was 4 definitions I have told. Diffusion, that is movement of molecules from high concentration to low concentration. Very good. What was osmosis? Movement of solvent from its higher concentration to its lower concentration through a semi-permeable membrane. Then I have told you about exoosmosis, movement of water outside the cell. And entry of water into the cell or movement of water into the cell is endoosmosis. Now, we'll understand certain thing about solutions and then this permeability, which I use the term semi-permeable. So first we'll understand the different types of solutions. So there are 3 basic types of solutions. Any idea? Hypotonic, hyper, and iso. Hypotonic, hypertonic, and isotonic. Now what is hypo, hyper, and iso? Okay. I'll quickly tell the meaning, then I'll ask you a very nice question, which I really love to ask. Okay. Hypotonic is a solution which has less solute. Okay. Just information that what is solute? Solute is anything dissolved in a solution. Okay. So if I'm dissolving sugar in water, so sugar becomes the solute and water becomes the solvent. Okay. Similarly, if I dissolve salt in water, so salt is the solute. A solution which has less solute becomes hypotonic. One solution, if I have more solute, becomes hypertonic. Okay. I'll tell first 2 only. The third one we'll wait. Okay. Now, a nice question. So the question is that I have a glass inside which nothing is there. This is not magic, sorry. Imagine there's a glass of water. I have added 20 spoons of sugar in it. What was this? Water. I have added 20 spoons of sugar in it. Now tell me whether the solution will be hypotonic, hypertonic, or isotonic. I know most of yours answer will be hypertonic. Okay. Do tell me whether you thought this or not. Okay. So, but this is not, you cannot tell. It's an incomplete question. How can we tell is, I need to have one more solution, not one more glass. I need to have 2 solutions to tell me which one is more or less. I cannot tell whether I am tall or short. I can be taller to someone and I can be shorter to someone, right? You have to compare my height to some other person. Similarly, for a solution, you have to compare it with the next solution. Then only you can tell. Suppose I said that 20 spoons of sugar and you said that it is hypertonic. Okay. I bring a next solution which is 25 spoons of sugar. So this is more or this is more? Of course, this one is more. So this becomes hypertonic and this becomes hypotonic. Let us suppose if these both solutions have 20 spoons each, then it is called as isotonic. So what do you mean by hypertonic? A solution which has more amount of solute as compared to other solution. A solution which has less solute as compared to other solution. And what is isotonic? A solution that has same solute concentration. 2 solutions having same solute concentration are basically isotonic. Okay, and just a hook, water always moves from hypo to hyper. Okay, now you will get to know why I'm saying this. Okay, now the next term, this is a membrane. Okay, imagine that this is the membrane. This side is a hypotonic solution. Okay. What is this side? Hypotonic. And this side is hypertonic to the same level. Okay. I've tried to put my hands on the same level. Okay. My nose, this partition, you can partition your screen into two parts. That is the membrane. Hypo, hyper. Okay. Water will move from hypo to hyper. Water will move from hypo to hyper. That is osmosis. Simple. The membrane should be semi-permeable. What is semi-permeable? Which allows the movement of solvent. A membrane that allows nothing to pass, non-permeable or impermeable. A membrane that allows everything to pass, that is freely permeable or permeable. A membrane that allows only solvent to pass, which one was this? Semi-permeable. And a membrane that allows solvent and selected solutes to pass through it is called as selectively permeable. Your plasma membrane or cell membrane that we have done, you remember this one, was or is selectively permeable. That means it allows the movement of solvent as well as certain selected solutes. So we have understood diffusion, osmosis, exoosmosis, endoosmosis, hypertonic solution, hypotonic, isotonic, right? And then we have done permeable, impermeable, semi-permeable, and selectively permeable. 11 definitions. One more term we'll do. Okay, the last term for this heading terminology, what I would like to say, explain is plasmolysis. Okay, very nice. Plasmolysis is just a kind of osmosis, but with specific, asterisk marks. Have you gone to sale and all? When we go for any brand sale, downside it is written buy, suppose outside buy one get one. Okay, on selected merchandise only. So it's like asterisk mark, conditions applied. Okay, now what are the conditions? Firstly, the cell should be plant cell. First, I'll tell the definitions and then I'll repeat the hook words. When a plant cell is placed in hypertonic solution. I hope you all remember hypertonic solution, which has more concentration. Okay. When a plant cell is placed in hypertonic solution, it will lose water by the process of exoosmosis. Ideally, it should have shrink, right? But because of the cell wall, it does not change its shape. And all the inner cell content move to one side. This condition is called as plasmolysis. And if I take this plasmolyzed cell and keep it in a hypotonic solution, it will regain its normal shape. I'll explain again. Imagine there's a cardboard box, okay, inside which balloon is there. Completely filled balloon, whichever was possible in that box. Now slowly, air is coming out of that balloon. What will happen? Will the shape of the cardboard box change? No, it remains same. But the shape of the balloon will change. It will start coming to one side as small as possible. That's the same thing that happens with the plant cell. It is just because of the cell wall. So what are the hook words in this definition? First is plant cell. Then which kind of solution do we use? Hypertonic. Then it does not shrink because of cell wall. Then what happens actually? The cell content, everything that is there inside moves to one side, and this condition is called as plasmolysis. So now we have understood many things about cell. So let's start discussing about the different parts of the cell. So of course, the first part is cell membrane. Okay, we have discussed about cytoplasm. So the next part that we are discussing is cell membrane. Cell membrane is the outermost covering of animal cell. I'll repeat, it is the outermost covering of animal cell. It is the second outermost covering of plant cell. That means what is present outside the cell membrane in a plant cell is cell wall. If you all remember or if you all know it, cell wall is only present in plant cell and it is absent in animal cell. Okay, so cell membrane is the outermost covering of an animal cell or the second outermost covering of a plant cell. It is made up of lipids and proteins. What is it made up of? It's very important to remember this. It is made up of lipids and proteins. Then the next thing is that there are many models for, plasma membrane, that how it must be, the lipids and proteins must be arranged. There's a very nice model. Okay. I'm a very foodie person, so I love food. So I'll remember that model every time. That's sandwich model. Okay. That means 2 layers of lipid is there and proteins are embedded in between, like a sandwich, but it's not like that. Okay. So the most accepted model for plasma membrane is fluid mosaic model. FLUID, that's fluid mosaic, MOSAIC model. Now, what do you mean by fluid? Fluid is anything that can flow, And mosaic means scattered, okay, which is not an ordered arrangement. Basically, that is scattered. So here in this, as per this model, fluid mosaic model, the proteins are embedded here and there. They are scattered here and there in between lipids. So the word mosaic. And why fluid? Fluid is the lipids give fluidity to the plasma membrane. That's why it can change shape, right? So that is fluid mosaic model. It was given by 2 scientists, that is Singer and Nicholson. They are teacher-student. Okay, very fascinating. Most of the 2 people, if you see, like Watson and Crick, who discovered DNA, the 3D structure of DNA, they are also a teacher and student. So yeah, so this is fluid mosaic model given by Singer and Nicholson. Now, what are the functions of plasma membrane? I will not tell. Okay, you only have to tell. First function was protection. Second, shape. Third, entry and exit of molecules. And fourth one was passing of message. Okay, so this was all about plasma membrane or cell membrane. The next part of the cell, very important, that we are going to do is nucleus. Nucleus is like brain of the cell. In our body, brain is the one that decides the functioning of our body. It controls, it regulates everything in our body. Similarly, in a cell, nucleus controls all the activities of the cell. Okay, so for this reason it is called as brain of the cell, control and coordinating center of the cell. Okay, nucleus, basically structurally, if I talk about, it has 2 membranes. Okay, it has 2 membranes which are not continuous, they are discontinuous membrane. Okay, and in this discontinuous membranes, inside is DNA. If I talk about a eukaryotic cell, it does not have naked DNA. So we say that it has chromatin, certain thread-like structures, chromatin. A dense body is there called as nucleolus. Okay, so this is about the structure of nucleus. I'll just repeat. What is nucleus? Nucleus is the control and coordinating center of the cell. Okay, structurally it has 2 membranes. Okay, so if I try and explain you, it is something like this. Now double all the membranes. Because diagram of nucleus is really important, it is usually asked by schools and exams. So this is the membrane of the nucleus. Okay, how many membranes you can see? 2 membranes, right? One is outer membrane, the other one is inner membrane. The gap between the 2 membranes, the gap between the membrane is called as pore. I'll just try and tell you. See, these gaps are basically called as nuclear pore. Then we have thread-like structure, nicely entangled thread, called as chromatin. This chromatin is made up of DNA and proteins. Which protein specifically? Histone protein, Histone. Okay, so I have told you about the nucleus structure, double membranous, pores, thread-like structure. Then it has a dense body here. Okay, this dense body which I have drawn just now is nucleolus. I'll repeat what it is. Nucleolus. What is the function of nucleolus basically? Nucleolus help in producing or manufacturing RNA, specific type of RNA, that is rRNA, ribosomal ribonucleic acid. I'll repeat, ribosomal ribonucleic acid. So quickly label these parts. This one will be outer membrane, inner membrane, nucleolus. Then what is this thread-like structure? Chromatin. Then what would be filled here? Nucleoplasm. Like cytoplasm is there for the cell, this is nucleoplasm. And the last one, what is this? Nuclear pore. Okay, so this is structurally a nucleus. Function of nucleus to control all the activities of the cell. So we have done plasma membrane, cytoplasm, and nucleus. Now we'll start with the cell organelles. So the first organelle that we are doing is mitochondria. You must be remembering, in your junior classes, we have learned and by-hearted this so much, that mitochondria is known as powerhouse of the cell. If you have not by-hearted, you have to by-heart it now, because it's really important. Okay, so mitochondria is again an organelle, very important organelle. Okay, it basically is the powerhouse of the cell. Now, what do you mean by powerhouse? That means it produces energy in the form of ATP. Okay, what is ATP? Adenosine triphosphate. Structurally, if we want to look about mitochondria, it is somewhat oval in shape. Okay, this I have drawn only single membrane. There are two membranes basically. Outer membrane is smooth, so I have drawn simply, but the inner membrane has numerous infoldings. You can see how am I drawing? There are many infoldings. These infoldings basically are called as cristae. What are they called as? Cristae. C-R-I-S-T-A-E. Okay. Outer membrane, inner membrane, and cristae. The entire inner membrane have lollipop shaped structure. Okay, like this. Lollipop. Yeah. So if you know a lollipop, it's like this. So imagine a microscopic structure of lollipop. There. Okay. So what are these lollipops? These lollipops are called as basically oxysomes. What are these called as? Oxysomes. Oxysomes are the basic ATP synthesizing unit. So if I say which organelle produces energy? Mitochondria. Which part of mitochondria produces energy? Oxysomes. So inside the packing material of mitochondria is basically matrix. And then it has a very important point for mitochondria. I'll draw it with another color so that you have an idea. It has DNA and what must be these dots? Okay, the somewhat circular shape I have drawn, that is basically DNA. It has a circular DNA, and the dots that I have drawn throughout mitochondria are basically ribosomes. Okay, so it has DNA and ribosome. Because mitochondria have DNA and ribosome, it is called as semi-autonomous organelle. Now, what do you mean by semi-autonomous organelle is that it can perform some of the functions on itself. See, mostly organelles are dependent on nucleus for their functions. Only two organelles are there which can, do half of the work by themselves. Out of those two, one is mitochondria. So mitochondria can perform few of its function on itself. That is why it is called as semi-autonomous. Okay. So three things we'll quickly remember. How many membranes does it have? Two. It has DNA and ribosome. For this reason, it is called as semi-autonomous. It produces ATP. For this reason, it is called as powerhouse of the cell. This is all about mitochondria. The next important organelle that we are going to do is chloroplast. Chloroplast is an organelle which is present only in plants. It comes in under the heading plastids. What are basically plastids? These are organelles found only in plant cells. That means they are absent in animal cell. Okay, plastids are basically of three types: leucoplast, chromoplast, and chloroplast. Leucoplast basically are colorless and helps in storing things. Okay, They store proteins, oils, etc. Chromoplast gives color, like you get nice color in the petals, green color, any color. Okay. Chloroplast, of course, gives you green color, so that means it's a type of chromoplast. But why do I categorize it separately? Because we are going to study it in deep, of course. The first reason. The second one is it helps in performing a very important thing or a process by the plant, that is photosynthesis. So three types of plastids: leucoplast, chromoplast, chloroplast. Chloroplast basically is a double membranous structure. Okay, that means how many membranes does it have? Two membranes. Now here the membranes are, both the membranes are smooth. So I have drawn two membranes. Okay, the textbook diagrams are usually 3D diagrams, so it's very difficult. So I always had an easy diagram of the textbook. So if you want, you may go with the textbook diagram also, or if you want, you may go for these diagrams also. Both are 100% correct, no doubt in that. Okay, so coming back to it, how many membranes does it have? 3. It has flat disc-like structure. Disc, you all know? Flat disc, coin-like structure called as thylakoids. What are they called as? Thylakoids. And stack of thylakoids. So if I put 10 coins one above the other, it becomes a pile or stack of coins. So this pile of thylakoids is called as grana. What it is called as? Grana. So thylakoids, one grana is connected to other grana by Fred's channel or stroma lamellae. So here are, so you can see many grana, many Fred's channel, like this. So this is somewhat the structure of chloroplast. Then what must be this? We have studied just now. These dots are, you guessed it right, ribosome. And the circular structure that I have drawn is DNA. So that means this, what was this? Chloroplast. Chloroplast also have DNA and ribosome. That means it also must be semi-autonomous. Very good. Okay, so I said that it gives green color. It gives green color because of the presence of chlorophyll. Okay, chlorophyll is present on the membranes of thylakoid. Chlorophyll is present on the membranes of thylakoid. So here membrane, this membrane, it is present on the membranes of, you can write grana or thylakoid, both are marked correct. Apart from all these structures, it has a starch grain. See, the final product of photosynthesis is glucose, but the storage form of food in plants is starch. So if the food has to be stored, it will be converted to starch. Starch is again made from glucose only. It's a polymer of glucose. So it is stored in that starch grain. This is the structure of chloroplast. I hope it's all clear to you. So two things that we have understood from mitochondria and chloroplast. If the question comes, name any two organelles that have two membranes or double membrane. So it is mitochondria and chloroplast. Name any two organelles that are semi-autonomous, mitochondria and chloroplast. Name any two organelles that have DNA and ribosome. Again, mitochondria and chloroplast. Next we'll do is lysosome. Lysosome is a very simple organelle. Lysosome basically has a single membrane. Simple. This is the diagram of lysosome. You'll say, ma'am, what you have done? You have just drawn a circle. It's something like that only. It has single membrane. Inside it is filled acid hydrolase. If you don't remember, if you find it difficult to remember acid hydrolase, what you can write is any digestive enzyme. So it has digestive enzymes in it. So what will be the function of lysosome? To help in digestion, right? The second and very important function, that it prevents infection from spreading. How? Suppose there are 10 rooms here, okay? This one room is there, and I am the lysosome, and some microorganism attacks to this room or to this cell. as a lysosome, what is my job? I'll burst. I'll kill myself, right? That is what is suicide. That lysosomes kill itself and release digestive enzymes. These digestive enzymes will eat the entire cell. The moment they eat the entire cell, that means they eat the bacteria or the infectious agent also that entered the cell. So with this, one cell of course was sacrificed, but the infection did not, moved to other cells. So this is how it prevents infection from spreading. This is about lysosomes. I'll just quickly repeat. Single membrane, what is filled? Acid hydrolase, if you can remember. Otherwise, you can write digestive enzyme. It is called as digestive bag because it helps in digestion. It is called as suicidal bag. This is very important and really important. It is called as suicidal bag because it, at the time of infection, it bursts and eat the entire cell. So it prevents infection from spreading. Next organelle that we are going to do is ribosome. Ribosome again are really easy. They are not very difficult. Ribosome are present in prokaryotic and eukaryotic both. Just a simplified diagram I would like. This is somewhat like ribosome. Okay. So you can see one part is bigger and other part is smaller. The bigger part is basically the bigger part that you can see is larger subunit, and the smaller part you can see is the smaller subunit. Now if I divide this structure into two, okay, why to divide, I'll draw one more. Okay, so this is one ribosome. This side is prokaryotic and this is eukaryotic. Okay, so in prokaryotic, this one, the largest subunit, name is given as 50S, and this smaller subunit name is given 30S. Similarly, for eukaryotic, the largest subunit name is given 60S and smaller is 40S. Now you'll say, what is this 60, 40, 30? What? Okay. See, a larger subunit and a smaller subunit. Understand that this is the largest subunit and this is the smaller subunit. They both join. They both join to form. This is the largest subunit and this is the smaller, I am saying. And they both join to form an ribosome. Okay. Now, when these both fall, they will go with speed, some speed. This and this fall separately. Will they go with, suppose this is speed X, this is speed Y, and when I join, it will be X plus Y? No. It will be a third speed, Z. Okay. Similarly, when smaller subunit of eukaryotic ribosome was sedimented, okay, by a scientist called as Swedberg, this went down with 40S for eukaryotic, I'm telling. This went down with 60S. But when these two combined, these don't say 100. It went down, I said, with the third speed, that was there. In this case, it is 80S. So just remember the values for eukaryotic. Then how to remember for prokaryotic? Do -10 from each value. So if eukaryotic largest subunit was 60S, prokaryotic largest subunit was 60 minus 10, that is 50S. Smaller subunit here was 40S, so this will be 30S. If this entire ribosome was named as 80S, so this entire will be named as 70S. Okay, easy? Right. What is the function of ribosome basically? Function of ribosome is to synthesize protein. For this reason, they are called as protein factories. Okay. So the question could be, which organelle is known as protein factory? Ribosome. In ribosome, there's one very important point. Ribosome is a non-membranous organelle. What diagram I have shown you is just a representative diagram to understand. It ideally does not have any outer membrane, a specific membrane or a bound. Ribosome is a non-membranous organelle. That means it does not have any membrane. The next organelle that we are doing is Golgi apparatus. It has many names like Golgi body, Golgi complex, Golgi apparatus. You can pronounce it as Golgi or Golgi, no problem. I would rather go with Golgi. Okay, so Golgi complex or Golgi body or Golgi apparatus is basically single membranous structure. Okay, it also has single membrane. Now this, it's something like this. Now this is the main body. Then it has vesicles around it. So the major structural organization of Golgi, if I can say, is cisternae and vesicles. The circular structures that you can see are the vesicles, and the flat disc with the curvature is cisternae. Okay, so this is cisternae and vesicles, the basic structure of Golgi apparatus. Golgi apparatus, basically the major function of Golgi is packaging and dispatching of materials. It packs and dispatch. Dispatch means sending it to other places or other organelles. Then it helps in secretion. So which organelle performs the function of secretion? Golgi. And then it helps in modification of lipids and protein. What does it do? It helps in modification of lipids and proteins. The next organelle that we'll do is endoplasmic reticulum. Achha, one thing. Why do you know, why did we give the name Golgi? Because it was discovered by a scientist whose name was Golgi. Camillo Golgi. Okay, so the next organelle I said that we'll do is endoplasmic reticulum. It is also a single membranous organelle, but it has certain dissimilarity with, if I say, with Golgi. So what I am drawing as of now is again cisternae, but here the cisternae are connected to each other. You can see this passage, they are connected to each other. Okay, and the cisternae, again beside the cisternae there are, what must be this? Vesicles. Okay, it do have other structures that support its structural features. Now on the membrane there could be ribosomes. What was the function of ribosome, if you all remember? The function of ribosome was protein synthesis. So if ribosome is present on endoplasmic reticulum, we call it as RER. Full form, rough endoplasmic reticulum. And if ribosomes are not present, we call it as smooth. That is SER, smooth endoplasmic reticulum. So there are two types of endoplasmic reticulum, SER and RER. SER does not have ribosome, that's why they are smooth. Then the second point we can write for this is they help in lipid synthesis. Okay, and they also help in detoxification of drugs. Now, what do you mean by detoxification? Toxic means poisonous. So it converts poisonous substances into non-poisonous, the drugs that we take, okay, the medicines and all. So it helps in detoxification of drugs in our body, removal of them. On the other hand, RER is rough because it has ribosome. And because it has ribosome, so it will produce protein. So if you remember, ribosomes were also called as protein factory. Similarly, RER also produces proteins. Now one doubt. If the question comes, which organelle produces protein? Will you write RER or ribosome? Always write ribosome. Okay. If they have to ask specifically for endoplasmic reticulum, they will mention in the question. So our next topic is vacuole. Vacuoles are again a single membranous organelle. They are found more, or you can say prominently in plant cell and less prominently in animal cell. What we can say is they are large and, you know, little in number or less in number in plant cell, and either absent in animal cell or if present, they will be very smaller in size and numerous. What is the function of vacuole? They help in storing. They are the storehouse. Now, if you have a storeroom in your home, it has all useful things, waste like old newspapers, maybe new shoes, anything that has been purchased must have been kept there. So store basically is for useful as well as for waste material. Similarly, vacuole is storehouse of the cell. So it stores useful as well as waste material. It is single membrane, or you can say unimembranous. Next is cell division. See, we have studied everything about cell. Cell, its different parts. And Rudolf Virchow also said that all cells arise from pre-existing cells. This happens by a process called as cell division. Cell division is basically of two types, mitosis and meiosis. I'll repeat, mitosis and meiosis. You just have to remember two things for mito and meo. Mitosis, from one, two new cells are formed. From one, how many new cells are formed? Two. Whatever amount of DNA is there in this, these both have the same amount of DNA. I'll repeat, mitosis, from one, two new cells, or you can write two daughter cells are formed, and the amount of DNA remains same. On the contrary, or on the other hand, meiosis, the second type, from one, four daughter cells are formed. From one, four daughter cells are formed, and whatever the amount of DNA this one has, all four have half DNA. Okay, if you want the process of mitosis and meiosis, I'll make a separate video for that. But for now, this is, more or less, this much is enough for you. Mitosis, from one, how many new cells? Two, and DNA remains same. Meiosis, from one, four, and DNA reduces to half. Very simple. In the end of this video, I'll ask you a question, and do answer me in the comment box and see. Let's see how many of you get it right. Okay, so this is about cell division. Now we have some differences to understand. That is animal cell and plant cell, and then prokaryotic and eukaryotic. So first we'll go with prokaryotic and eukaryotic. Prokaryotic basically does not have a well-defined nucleus. That is what the name is, prokaryotic. Pro means primitive, karyo means nucleus. So they have primitive nucleus, means well-defined nucleus is not there. Now, what do you mean by well-defined nucleus? Nuclear membrane is absent. What is absent? If you remember the double membrane with nuclear pores I have drawn, that is absent. Rest everything is there. Eukaryotic, EU means true. What does it mean? True. A true nucleus is present. So in this, a well-defined nucleus or a true nucleus is present, which basically means nuclear membrane is present. Second difference. Prokaryotic does not have membrane-bound organelles. That means all the organelles that I have drawn with a membrane, like one membrane, two membrane, are not there. But in eukaryotic, all are there. Okay, I'll just quote a few examples that mitochondria is not there in prokaryotic, chloroplast is not there, lysosomes, vacuoles, all this is not there. Okay. Then comes that ribosomes. If you remember ribosomes, we have done for pro and eu. Ribosome was of 70S, and in eukaryotic, ribosome was of 80S. Prokaryotic is generally smaller in size. On the other hand, eukaryotic is bigger or larger in size as compared to pro. Prokaryotic have only one chromosome. Inside its nucleus, only one chromosome is there. But in eukaryotic, there are many chromosomes. Prokaryotic DNA is not bound to histone protein. You remember I told you that in chromatin, histone plus DNA is there. So here histone is not present. DNA is all alone. But here DNA is present with histone protein. These are few of the differences between prokaryotic and eukaryotic. Example of prokaryotic is bacteria, and eukaryotic, you can write anything. Human. Okay, you can write other animals as well. Then we have difference. The last topic is difference between plant cell and animal cell. Plant cell is basically the basic differences. They have a cell wall, and animal cell does not have a cell wall. Okay, does not have a cell wall. Plant cell have chloroplast or plastids. Animal cell do not have plastids. Plant cell do not have lysosome. Lysosome, you remember, na, suicidal bag or digestive bag. Animal cell, on the other hand, have lysosome. Plant cell, vacuole, a very big or large vacuole is present, usually one in number. But in animals, Small size vacuoles are present and there are many in number. Now, because of the vacuole in plant cell, the nucleus is shifted to periphery, or you can say corner. But in an animal cell, nucleus is present in the center, right? So nucleus is at the corner, nucleus is at the center. Vacuole, large, one, vacuole, small. One more point we'll do, centrioles. Centrioles are the one that help in cell division, okay? Centrioles basically are absent in plant cell and centrioles are present in plant cell. One very difficult thing I'll leave on to you only, I will not tell. Okay, you have to write 2 examples of plant cell and animal cell. That means you have to write 2 examples of plants and animals. So here we are done with the fundamental unit of life. Hope you have liked the video and understood the concepts of fundamental unit of life. If this video was helpful to you, please do comment and let me know. All your suggestions are always welcome. And for the latest update, please subscribe to my channel. Thank you and take care. Bye.